First, welcome everybody. My name is Mohammed Iskandarani. I'm a professor in the Department of Ocean Sciences, and I've been coordinating the IDSC Fellow Program for this year. I would like to thank all the applicants and especially the recipients of the fellowship program this year, Sadegh, Kelly, Jeffrey, and Shara, who's joining us remotely on Zoom. I have some special thanks for the steering committee as well, who helped sort out the applications and choose the fellows. Just a few words about the program before we start listening to the presentations of the fellows. What is the project, why, and how, mostly. The program was started about more than 10 years ago to sort of encourage interdisciplinary research across the units of the University of Miami. Scientific computing now permeates all scientific endeavors and the humanities. And we thought CCS, Center for Computational Science, and then later on IDSC, to be the perfect place where the disciplines should meet and leverage each other's expertise. Okay, so in this iteration of the program, we usually elect 4 fellows, 2 graduate and 2 undergraduates, but this year the committee decided that to choose 3 graduate students and 1 undergraduate from the different units in the university. So, Nick, director of the program, would you like to say something?
Well, if you put me on the spot, yes.
No.
Over there. So first of all, I just wanted to welcome everybody. Maybe I should use this, right? So I wanted to also welcome, can you hear me on the Zoom?
Shara, well, why don't you stand here on the podium? Oh, the microphone is muted.
Okay, thank you. Okay, so thank you everyone for coming here, both joining us from Zoom and in person. So this is so first of all, I want to thank also all the applicants and of course our current 2022 IDSC Fellows, 22-23 IDSC Fellows for their hard work. I also want for putting the application together and becoming our this year's IDSC Fellows. I also want to thank the committee and Dr. Iskandarani, who chaired the committee this year, for their great work, because it's this program, as Dr. Iskandarani was talking, was actually interdisciplinary. So he's trying to bring disciplines together, and for that's why every one of our fellows has got a dual mentorship from 2 different fields that they are where the projects require expertise that they span or they are in the interface of 2 different fields. So this is a very unique program that we've been doing this for many years, but also this is expose our students of how this intersection between different fields can happen and how much progress you can make in this interface and what are the catalyzing factors and innovations. As a result, though, our committee had a very hard task, because not only had to select outstanding students to become fellows, but also select the mentors. So they had to do a matchmaking, if you will, between faculty members, the fellow interests, and in order to understand this kind of interdisciplinary approach. So that's always very tricky. but it's always has produced over the years some amazing results. Not only that, the experience for the fellows or exposed to this interdisciplinary approach, but also sometimes you see that the mentors are continuing their collaboration beyond and above, this duration of the fellowship for the students. So welcome everybody. Looking forward to hear your presentations and looking forward to this year's IDSC Fellows.
Thanks, Nick. So without further ado, let's hear about the project. Jeffrey, would you like to come out first?
All right, so my project is on orientation selective deep brain stimulation of the midbrain circuits that control locomotion to improve gait following spinal cord injury. The project overview is to overcome computational bottlenecks commonly encountered in like traditional desktop or workstation environments for specifically pre-processing diffusion weighted images. This can take upwards of 16 or 8 to 16 hours. And it's a huge bottleneck in our computational workflow. Also, we want to improve DBS outcomes by offering more realistic visualization. And overall, our ultimate goal is to decrease the time it takes for DBS therapies to go from bench to bedside. This is an example of the experimental setup. So we set up the pigs with intramuscular EMG, and we record local field potentials from the DBS electrodes in both the left side and right side of the brain. Our computational work, you can't really see it on the side. But anyways, this DWI FSL preproc is the command that specifically takes about 8 to 16 hours to do. And by uploading MR-Trix, this parallelized program to the supercomputer, we can decrease this time to roughly 30 minutes to an hour. And this is a simultaneous process that's co-registering a template micropig brain to co-registering the DWI patient space to the template brain. So we all work in the same space in order to do group-wise analysis as well as patient-specific analysis. Sorry to interrupt. Yeah. You're good. There you go. Here's an example of a tractography computed from the previous computational workflow that I presented. This specific tractography is seeded by the MLR, the mesencephalic locomotion region in the brain, pointed right here. And here are some, oh, wow, that is not supposed to be there. These are some preliminary results of the tractography generation, and you can see the isosurface is color coded based on this scale right here, 2.5 to 0.61 millivolts. And here's an example of the intraoperative and treadmill-oriented recordings of EMG, and the same stimulus parameters set in the computational workspace. And here the vector gradient potential is shown in arrows. I know it's hard to see. But here are the colored fibers that are activated. And this is what we're really trying to tell from this computational workflow is what percentage of fibers are activated versus those that are not, and trying to optimize the activation of fibers that produce locomotion versus those that produce side effects.
Where is the probe in the picture?
The probe, the electrode,
Yep.
It's right here. This is the electrode. Sorry, I skipped over this. The electrode design is a Boston Scientific directional electrode, which has been commonly used to steer current in the brain based on its position and these directional contacts. These are the contacts that stimulate via current or independent current induced stimulation. They are the ones that can either project this way, that way you can't see it, or this way. And there's 8 of them in a 1-3-3-1 configuration. And this right here is the directional marker in order to see on the postoperative CT where the electrode is placed in relative to the rest of the brain structures. Yeah. Blue is activated, sorry. This color map is only for the isopotential, which I made a mistake here. There's no isopotential being shown here. So this really color map doesn't mean anything right here. And that's it.
Thanks, Jeffrey.
Questions?
Over 8 hours.
16 hours.
16 hours.
Wait, is it 8 hours to 16 hours? I just wanted to clarify, sorry. For that one process within the entire workflow. It's in order to denoise and eddy current distortions, as well as motion induced distortions that are produced in diffusion weighted imaging. No, but I didn't get to that. Sorry. I do want to implement some deep learning segmentation within the workflow, because we have a pig brain atlas that is on paper, but we can use this in a computerized form to segment brains in an automated way. Yeah.
You usually know which nerves you want to.
Yes. So we target the cuneiform nucleus, which is around 5 millimeters lateral to the midline. Yeah.
And once you segmented it, do you repeat the calculations, or is it,
Usually we just use one tractography and compute the finite element mesh in response to the point source current.
Thank you. Want to go next? Right. Our next speaker is Kelly Soluri. She's a Master of Science students in marine biology and ecology.
So my name is Kelly Soluri. I'm going to be presenting on forecasting bycatch hotspots using machine learning. And so first, let's cover what's bycatch. Bycatch is untargeted species. So when fisheries go out, they normally have a target species that they're looking for, let's say tuna, and everything else that they catch that isn't tuna, non-species, or let's say the tuna is too small to commercialize or it's spawning, they can't use it. All of that's considered bycatch, so it's wasted. And so bycatch has really long-term negative effects on not only on, it impairs species conservation efforts in the case that it catches endangered species like sea turtles. It can disrupt food web ecosystems in the case that it catches like a top predator or a keystone species like a shark. And it's also economically wasteful. It costs the commercial US fisheries millions of dollars a year. So it's kind of a lose-lose situation for everybody. The fishers want to fish the right fish. The fish don't want to get caught. So we want to go for the right fish and not catch the wrong ones. And so we want to try to forecast these ecological hotspots in a better way to mitigate them by avoiding them. And so in the past, ecological forecasting has occurred in the sense that they look for a single bycatch species, like a type of grouper that we don't want to catch if we're a fishing tuna fishing boat, right? And so they use a linear regression or a GAM, a generalized additive model, using this singular species in order to better forecast where they'll be and try to avoid that. And in newer ecological forecasting, the image to the right is showing this type of ocean dynamic management, which uses habitat predictive models instead to better predict for bycatch. So in this case, this paper and Hobday was looking at Australia and predicting these species where they would be based on what kind of habitat that they prefer. And that was a lot better than the previous one. Now, what I'm going to be trying to do is not only not make it single species, we're going to try to use multi-species, and we're going to be using machine learning instead of habitat predicting or GAMs or any type of other linear regression. And to the left, this is the amount of bycatch intensity in the world. And you can see that it's pretty high around the Americas. And if you try to look in Florida, you can see a couple different colors. So that makes that the Gulf of Mexico and around Florida a really ideal study spot for us to look at this kind of interaction, because the Gulf of Mexico has an incredibly high biodiversity. It's the area of the body of water in the United States that has the highest biodiversity. And yet the amount of multi-species studies is very minimal because it's hard to cover so many different species. And so we're going to be trying to look for these kind of multi-species interactions and using these multi-species complex interactions to better forecast them. And we're going to be using the long line fisheries in order to be the fisheries that we're going to be doing this case study on. And they normally fish around the shelf. So if you look at the Gulf of Mexico food web to your right, the shellfishes in the benthic community on the shelf is a transitory zone. So you get both the pelagic deeper species, so the species that are more in deeper waters going up to the shelf, as well as nearshore and juvenile species going out into the pelagic ocean. And so you have a lot of overlap in this shelf area, as well as an overlap to where the fishers want to fish. And so that makes it an ideal fisher community for us to base our data on, as well as a good area to look for fish abundances. So there's a problem in our data set. So we get these kinds of data from this program called the Observer Program by NOAA. And so they get these people called observers to go on these commercial vessels, and they measure these fish that are caught as bycatch, as well as environmental data of where they're fishing, what kind of activities happening with fishing, what gear they're using, how long they're fishing for. And that's all data that we use, except it only covers 8% of our fleet in the Gulf of Mexico. And so there is an imputation issue there. And as well as the reasons that we want to look at the Gulf of Mexico is that it's highly diverse, but for that exact reason, it's also an obstacle for us to overcome with machine learning, because each fish has its own requirements, quirks, biological traits that we also want to help predict in the model. And then it's also multivariable. So the bycatch problem is split into two parts. You have the anthropogenic part of the problem, which is the fishers, how they act, their behavior, where they want to fish, how long they're fishing. All these efforts is one half of the problem that we try to predict. The other half is the actual fish abundances. Where are fish likely to be? How much are they going to be there? How biodiverse is this spot? So it's a multivariate problem. And we were looking at different tools to be able to overcome these issues. And we thought that a random forest model was a pretty good tool for us to look at, because it deals with a lot of these problems in this way. So one, it's a powerful predicting and imputing tool. And in other studies in the past, like they put different GAMs and different predictive models, and random forest turned out to be very good, even with the lack of data that was going into it. And so that's one really good reason why we're going to choose that. With highly diverse with highly diverse data and multivariable data, it's also very flexible, and it's able to take different samplings of both the variables and the predictors. And so it still makes it very statistically powerful, even though we're using different types of data from different sources. And so that's why we were looking at random forest to be our machine learning. And so our plan is to do the training data based on 2/3 of our data set. And so here on the left, we have a map of the current reef fish bycatch hotspots in the Gulf of Mexico that I've mapped up. And then as well as where the current vessel densities in the Gulf of Mexico are. And we're going to take 2/3 of this data to train our model, and the last third to do our out of bag estimates and to show the accuracy of our model. And so what we're looking for in methodology, first of all, we're going to test the levels of correlation in our anthropogenic and environmental variables. And then we're going to test these different variables in different models using different algorithms using MATLAB. Then once we decide on a particular algorithm and model, we're going to train it using those maps showing you the data, what that looks like. We're going to train the model using that data to predict catch per unit effort, which means just how much fish is going to be there per the effort of fisher in that area under different conditions. And then finally, we're going to use an information criterion, or a Kaiki's information criterion specifically, to determine accuracy and compare that with previous forecasting attempts. So I'm really happy to have this team behind me, helping me and guiding me in this project. I'm really excited to learn more about new ecological forecasting methods and novel methods that haven't been tried before. I'm being mentored by first my PI, Dr. Elizabeth Babcock, who's a quantitative fishery scientist here and a professor here at Rasmus. And specializes in fishery stock assessments, providing me the background that I need for the ecological portion. Dr. Gang Ren is my IDSC mentor, and he's a data and computer scientist here at specializing in artificial intelligence. And finally, Dr. Mitsunori Ogihara, who also is a really big person here at IDSC, was a director of development, workforce development education with research interests in data mining. So I just want to acknowledge first IDSC for choosing me. I'm very excited. When I heard the news, I was very, very happy. And I'm so excited to get started on this project. I also want to thank my funding, which is the Cooperative Institute of Marine and Atmospheric Studies, or CMIS, and NOAA. And with that, I'm ending my presentation. I'm ready for questions.
Any question for Kelly?
So the data you said comes from the ship GPS?
No, it comes from the observers. They go out. These people are called observers. They're under the observer program by NOAA. And these are actual people who go on the boats as a worker. And instead of fishing along with them, they just record all the fish that are caught up that are wasted. So that's where we get our data. And it's spanning many years, over 20 years of data.
The random forest model?
Yes.
Is that like a GPU intensive model, or?
A GPU, could you just break that down, what that actually means?
So like will that be run on a compute like CPUs, or would that benefit from running like on graphics like processors, like on Triton, for instance, where we have with the GPUs, you have a lot of cores that can run like vector kind of mathematics really quickly compared to computation, where it's just a CPU, which is a lot slower because you have less cores. But I should be familiar with the random forest, but I'm not.
No, it's really exciting. I actually didn't know about that. And one of my concerns was that since it is intensive, like what I was going to do with like all of whether I was going to get a blockage in time or something like that. I was told that MATLAB is a good platform for me to do the random forest that would be faster. But if you know of any other ways that would be helpful, I'd love to listen and talk more in depth.
I'm definitely going to look into.
Thank you. Yes.
Based upon 20 years of data?
Yes.
What's the approximate size of the data set?
It's around the actual number. I don't want to misspeak.
Just approximation. It doesn't have to be accurate.
I'm going to say like 10,000.
per sample or?
No, the whole thing.
Oh, that's not very large.
Yeah.
Yeah.
How many classes?
How many classes of different species? There's a lot of different species. We have our most popular species right now, which is red grouper, followed by yellow edge grouper. And those make up a large percentage of our bycatch, but we also have a lot of many different types of species that make us a very small portion of our bycatch. It could be anywhere from, I think, around like 20 to 50 species. Because we're also not only looking at reef fish, but we're also looking at sharks and sea turtles.
Do you plan to like grouping these together?
Yeah.
5 large groups. Subclasses.
For the forecasting, I hadn't considered it in that sense. I felt like putting it in one would allow since the random forest already randomizes it, that there wouldn't be much of a point, but I'd love to hear like what you think the benefits are, and I might change that in my methodology. It's in the beginning, so I'm open to all comments.
I have a question.
Yes,
Do
please.
fish vary seasonally in their position?
Yes, they do.
How
They-
how do you accommodate for seasonal variation of the different species?
Mhm.
the
So that's gonna go in, so that's two parts, because not only do fish move, fishers also have their own behaviors. And that's a really, really good question. In my actual dissertation, I have a model after this one that accounts better for fisher behavior and fish behavior. And that was gonna be how I was gonna deal with that. But within this problem, I'm not actually sure how we would deal with that. And I'm open to hearing any suggestions.
Are the fishers, are they kind of more locked into a geographical location, given that they have a port that they come out of and have a certain radius that they're kind of locked into?
Yes, very much so. So
You have some way to restrict their area of coverage.
yeah, so if they're coming out of Texas, they're not going to show up on the West Florida shelf. If I can go back to this slide here, most of our vessel density is on the West Florida shelf. But we're also most likely where there are the most fishers are going to be where there's historically most fish. And in the West Florida shelf, there's a really big fishing community because of that. The shelf is much larger. And so there is somewhat of a depth component, which is a predictor variable that's very significant. And so these more of the environmental restrictions are more powerful than like the actual like whether they're coming from Texas or Louisiana. You can see like a really big split in between of fisher behavior between the East Gulf of Mexico and the West Gulf of Mexico. But I think that environmental variables are more restrictive than the actual ports they're coming out of.
As a follow-up with like the Deepwater Horizon event
Yeah.
and the oil deposition that happened
Yeah.
and loss of marine life, how is that going to be taken care of in the data set?
Yeah, so that is a really big factor, not only with Deepwater Horizon, but also with COVID. That's one of our issues, right? Because the data needs to be imputed for these kinds of gaps in the data, which is what I was hoping random forest would be a good model for. So that's like, just like with the internal like mechanics of how that model is, that's kind of why I chose it to cover those imputations.
Any other questions? Maybe from the audience from Zoom? I haven't been monitoring.
Can I make a quick comment on this?
Yes, please. Who's speaking?
This is Mitsu.
Hi, Mitsu.
Hi. So let's see. So given the scale of the data and given that the random forest models will be explored. So I'm sort of answering the earlier question about the use of GPU versus CPU. So my impression is that given the scale and given the nature of the analysis, the use of GPU may not be so helpful. So I think that the choice of using a CPU and MATLAB, which is very handy to the University of Miami students because it's available to everyone, is the right direction to do to follow. Say after some exploration, if there is a need for using much larger scale for exploration for parameter search, probably we could use the Pegasus. That I'm not thinking not Triton. Probably Pegasus could be a useful tool in speeding up the process of exploration.
That's very helpful. I wish I had like a paper so I can write down what you just said so I can refer to it later, but we have the recording here and I would love to talk more in depth with you about where you think the right direction is for this project. Thank you so
Yeah,
much for your
be-
comment.
Be glad to.
Thanks, Kelly.
All right,
This-
thank
this
you.
was great. Sadegh.
Hi everyone, I'm Sadegh Tale. I'm from Department of Civil and Architectural Engineering. My project is segmentation and evaluation of microcracks from macro city images of concrete elements. So let's start at the very basics. What is concrete? Concrete essentially is made out of cement, water, and aggregates. This is the most basic concrete that you can have, but realistically, concrete has a lot of other stuff. For example, a lot of chemical admixtures, different fibers, and steel reinforcements. Concrete has been a turning point in our construction industry, has enabled us to build a lot of massive infrastructure like dams, bridges, has provided a lot of housing for a lot of population. But of course, it has huge negative impacts on the environment as well. For example, on the left, you see the greenhouse emissions of concrete related, divided by sector, and you see the building sector is the third place in terms of emissions. Also, it makes up a huge chunk of the transport. So it emits a lot of carbon dioxide and other methane, for example. I think the estimates right now are 8 to 9% of total CO2 emissions. In terms of material use, you see sand, gravel, rocks, coal, limestone, clays. These are all materials that are used in cement and concrete production, and they make up a huge chunk of all the materials that we excavate. So concrete is great, but it also has a huge impact on the environment. If it was so that we could build a structure and it would remain for a long period of time, that would be great. But in practice, concrete deteriorates really fast. Sometimes the service life is around 50 to 100 years, but in practice, we see that buildings start to crumble between 20 to 30 years. And these are because different environmental conditions such as chloride attack, freeze-thaw, sulfate attack, and other environmental impacts can really deteriorate concrete structures. For example, chloride attack causes corrosion, which right now makes up 3 to 4% of total US GDP. Of course, all of that is not concrete, but a large amount is. So it is really important that we monitor our structures and prevent them to reach such conditions. The main property that facilitates these deteriorations is the transport of liquids that contain negative chemicals such as chlorides and sulfates into the concrete structure. So we want to mitigate this transport as much as possible. One of the things that facilitates this transport is the cracks that happen in concrete as time passes. And this is what happens if we don't monitor the infrastructure. On the left, we see Ponte Morandi Bridge in Italy that fell down in 2018. I think 40 people lost their lives. Or on the right, you see the Chaplain Towers. New York Times says that abundant cracking and crumbling of the columns was noticed, but they didn't do anything about it. So it is really important that we monitor these structures, both in terms of economical sense, but also human life. So there are a lot of methods to do this health monitoring. We have visual inspection. From a long time ago, people would go and look at the structure and see if there are any cracks or not. There are automated systems such as the drone drones that go around the structure and take pictures. There are automatic static and dynamic monitoring, for example, with sensors to measure the displacements, the temperature, and so on. There are a lot of methods. One of the methods is sample extraction or coring, which is basically a machine comes and takes a core out of a slab or whatever, and then they can run some tests on this core and find out what is going on with the entire structure. And on these core samples, a lot of tests can be run. One of them is CT scans. So this is a summary of how CT scans or micro CT scans work. So you have a sample, you put it in a machine between the source, X-ray source, and a screen. These X-ray beams come and hit the sample based on their density distribution and their shape. Some of these X-ray beams get absorbed, the other ones pass and hit the screen, which gives you an image like that. And next, you can turn the sample slightly, for example, half a degree, take another picture, and do that until you do a 180 or 360 of the sample. With some mathematical formulas and algorithms, you can build up a 3D image of the sample, which gives you not only the surface, but also the cross sections, so you can see everything that is going on in the sample. The difference between macro CT and CT scan is that CT scans are those that usually are used in hospitals, and they have very large resolutions. With macro CT, you can see objects as small as 2 or 3 micrometers. We have a micro CT in our lab. We've done a lot of collaboration with different groups to map different objects. For example, you see tooth here, human vertebra, skulls, corals, insects, flowers, fish, precipitates, and so on. So it is a huge and it's a valuable tool that we're using to do a lot of research. In terms of how it relates to my project, we have a lot of crack images on the literature and on the web, but most of them come from the surface. There are some papers that do CT scan on concrete columns to measure the cracks inside the concrete and not on the surface, but these are usually done with CT scans, which means that this crack that you see here might be in order of millimeters, and that is a huge crack. Usually when you have cracks that large in your structure, they have already sustained a lot of damage. So you don't want them to get to this level. The way micro CT helps is that you can map cracks that are very, very small, 5 to 10 micrometers. And at that stage, you still have time to go and do some repair or actually do something. So first of all, I have these databases. For example, I have around 12,000 images with their mask from different sources. I have some databases on these, but what I want to do is make up my own database as well with cracks that are in order of micrometers. For example, this is one of the samples that I've made. You see a lot of voids here. These black spots are the voids, and the white or the lighter spots are the concrete background. And this high amount of void is intentional with some kind of admixtures. I'm trying to build up a well-rounded database with different shapes. This is just a few trials that I did. So here you see a crack that is running here. This should be around 10 to 15 micrometers. Another one here. This is another sample. You see the cracks here as well. I'm gonna make some samples with aggregates as well, and with different, for example, void areas and so on, to have a well-rounded database. But this is just a few samples that I did. So so the goal here is to segment these cracks as accurately as possible, so we can actually measure the degree of damage in the structure automatically. And so let's say we have a few cores, we run this microcity on them, and then we can assess the degree of damage and the degree of cracks in them. People use a lot of different algorithms that I've seen in the literature. Most of them are CNN related. So for example, this paper used our CNN. What I plan to do is to look at different segmentation algorithms, for example, UNet, LinkNet, and so on. Combine that with some backbones like Inception and so on. This is just in the earliest stages. I haven't done any work in this area yet, except for a preliminary code, but this is what I plan to do. I'm hoping to combine that with transfer learning to actually train the model on these images, and then transfer that learning into my database, which will be smaller. I'm hoping for like 500 to 1,000 images, but we'll see. I did a preliminary code with UNet and I think ResNet on these images, not on my own. Like on the test data set, you see the accuracy is very good, and I want this because I want to also quantify these cracks. But in evaluation right now, they're not very good. The cracks, the masks that I produce are much thicker than in reality. So, I mean, this is just a basic code that I did. I need to do a lot of more work on this area. This is our team. Dr. Ali Ghahramani-Nejad is my PI and academic advisor. Me and Dr. Chapman is the IDSC mentor. Hopefully we can do some good work on this area. I'm doing some other machine learning work as well. So my PhD topic is mostly focused on chemical admixtures that we add to concrete. So I'm doing a lot of colloidal testing of proteins and polymers in different solutions. This is another project not related to this. I just wanted to mention, we're trying to make a link between the biomolecules and their molecular structure to their properties in colloidal solutions, and also their compatibility with different cementitious materials. So our goals is that hopefully first I can learn more about segmentation and transfer learning. I have some background in machine learning, but not on these areas. I have to make a database to actually include microcracks, and then hopefully we can build a pipeline to accurately segment and measure these microcracks. Thank you. This project and the other one is supported by NSF and an MRI award. Thank you.
Question for Sadegh. I have a question. So one of the challenges is to be able to distinguish between voids and microcracks.
Not only that, but also usually there are a lot of other stuff in concrete. For example, you have aggregates. You see here these aggregates. I'm gonna make samples with aggregates. Right now, this sample is only cement, water, and some chemical admixtures. But hopefully they can be able to differentiate between the voids, between the cracks, and the different aggregates.
And one more question in terms of, are you planning to quantify the actual crack?
Yes.
Right.
After
So,
segmenting them, I want to quantify them to have a metric of.
so the current goal is to segment first,
Yes.
right? Obviously,
Segment very accurate.
right. And then the measure. So are the actual, what is the actual, dimensions of the crack? So how would you?
I mean, you can get a lot of different metrics. I haven't looked at that area yet, but basically you can have the length of the crack, you can have width of the crack, or you can have the area.
That's what I was getting
ratio
into,
of the
right?
sample. Yeah. Something like that.
When you get these core samples, how large are the samples that you're getting?
It depends on your structure. If you have something like a dam, you can take large cores, for example, 15 centimeters in diameter or possibly larger. But with buildings, you cannot get those huge cores. But what you can do is you can have thinner cores, but with more frequency from different parts.
And are you planning to correlate any of this with maybe stress strain transducer measurements and with the cracks and
you
the
mean-
micro cracks that you might have?
Transducers,
So that you have it.
UPV and that kind of.
Yeah, so you have some way to measure the stress strain put onto it. So you have some machine that could actually strain the sample
Yeah.
in some manner, and you get a measurement of that force it takes
Yes.
to cause deformation, and then relate that back to the cracks that you have identified.
So these samples that I made, I put them under compression to cause these micro cracks. I can possibly measure those displacement or forces and correlate them later. I haven't thought about that. That should be something to think about. Thank you.
This is really out of my territory, but so I would imagine if you're doing a core sample that puts a lot of stress on what your decore or your coring. So is there a way of dating a crack and to know whether or not the actual coring process is actually causing a microfracture?
What is the question again? I'm sorry.
You don't know if the core sample you took is the microfracture was caused by the fracture you actually recorded the sample.
Oh,
Versus graded that out, or if it was there prior to that process.
I don't have that much experience with coring, so I don't know. I'm not sure. Yeah.
Great presentation,
Thank you.
Sadegh. I may have missed this, but I was wondering, I know that concrete, there's like a lot of different things that it's made up out of. I was wondering if you were using the same mixture to test the micro cracks, or if you were using different types?
So I plan to change the variables, as I said, to have a well-rounded database. So some samples are only with cement and water and some admixtures. Some will include coarse aggregates and fine aggregates. They will have different admixtures, different. So I'm trying to like change the variables to like have a diversity in the database. Thank you.
Thank you.
All right. Last but not least, our undergraduate fellow this year is Shara Sookhoo. And let me see, let me stop the sharing. Shara, can you hear me?
Yes, can I go ahead and
Okay.
share?
Okay, so Shara is an undergraduate in marine biology and ecology, right? And she's working with Nikki Traylor Knowles, and she would like to build some sort of machine learning models to study the microbiome of coral reefs, right? So you can go ahead and share your screen and give your presentation.
Hello, can everybody hear me and see the presentation?
Yes, we can hear you fine.
Okay, great. Okay, so as the professor said, my name is Shara. I'm a junior majoring in marine biology and ecology. And my project is to investigate the coral occurrence patterns in microbiomes of 6 species of corals. There we go. So coral reefs are biological hotspots. What that means is that even though they account for 0.5% of the ocean seafloor, they contribute to more than 1/4 of our marine biodiversity. To put that into perspective, that's 6,000 species of corals, supporting 4,000 species of reef fish and 1 million species of other organisms. Of those thousands of species of corals, 800 are what we call reef builders. These are the corals that deposit calcium carbonate to build their skeleton and therefore make up the stony framework of the reef. This stony framework dissipates up to 97% of wave action and therefore protect 15,000 kilometers of shoreline from erosion, amounting to a $4 billion value. Without reefs, the damages from flooding is expected to double, and the damages from storms is expected to triple. The stony framework also provides a habitat for organisms to live and thus attracts the biodiversity to the reef. In countries such as the Maldives, reef-based tourism can contribute up to 64% of their GDP, and internationally, reefs contribute to $36 billion. In addition, reefs are an important source of protein and provide livelihoods for 6 million fishers, amounting to $6.8 billion. The sad truth is that reefs are under threat, and it is expected by the end of 2050, 90% of coral reefs will become functionally extinct. The main threat to coral reefs is climate change. This is one of the most famous pictures of coral bleaching. The picture on the left was taken in December 2014, and the picture on the right was taken in February 2015. In less than 3 months, a healthy reef was completely bleached and died. In healthy corals, there's this photosynthetic symbiont called zooxanthellae, and they provide the characteristic color of corals, and more importantly, the food for corals through photosynthesis. But under stress, those corals release the zooxanthellae. They lose their color and they lose their source of food, becoming bleached. Some corals may regain their zooxanthellae after a while, but most don't. And essentially, they starve to death and die. This is the coral bleaching map from NOAA for today. And the area encircled in the black is the coral triangle. It is home to 76% of all reef-building species of corals in the world. And right now, it is under coral bleaching warning, alert level 1 and alert level 2. Warning means that coral bleaching is possible. Alert level 1, the red, it means that coral bleaching is significant. And the maroon, alert level 2, means that coral bleaching and coral mortality is highly possible. This means that as I speak, these reefs off of Papua New Guinea in the coral triangle are bleaching and are most likely dying. Following bleaching events, disease often occurs because pathogens take advantage of these susceptible stress corals. In the past 40 years, the incidence of disease has increased both in terms of prevalence as well as geographic distribution. But not all hope is lost. All around the world, here at home and internationally, a lot of people are doing work to restore coral reefs, even here at UM. Conservationists take advantage of corals' ability to reproduce asexually. What that means is that fragments are taken from adult colonies and then grown in a land-based nursery or sea-based nursery to some threshold size. It depends on the species. And then when they reach that size, they are outplanted onto degraded reefs by attaching it to the existing substrate. It is hoped that these fragments will eventually become healthy adult corals to support new reefs. But the sad truth, again, is that these corals fall victim to the same threats facing the existing naturally occurring colonies. So why do we bother? Well, in the words of Dr. Lerman, who is the founder of UM's Rescue Reef Program, if we do nothing, then there will be no more reefs to save. Because if we sit and wait for those policies to be put in place to change the courses that are affecting reefs, then by that time, there'll be nothing left. But it is difficult to identify these resilient corals to outplant, so we could buy more time. The coral holobiont refers to the host as well as the symbionts, the viruses, the archaea, the fungi, the bacteria, and the zooxanthellae. And any change in any one of these symbionts or the host itself could affect the resiliency of the coral. My study will specifically focus on the bacteria. Why the bacteria? Well, there's been a lot of research going on recently on the microbiome because it has been found to confer resilience to bleaching, to disease, and nutrient loading. And recently, we've been able to prove that there are certain species of bacteria that act as the pathogens that cause coral diseases. However, the microbiome is difficult to study since many of the coral associated bacteria cannot be cultured in a lab. This study therefore takes a genetic approach to identify and quantify the bacteria found in these 6 species of coral. These are the main species housed at Moats International Center for Coral Reef Research and Restoration, where I interned over the summer with the Coral Health and Disease Lab. There, I would have sampled 30 genotypes of each of the 6 species and then processed the tissue to extract the total DNA. The bacteria would have then been sent to a third-party Mr. DNA, where the bacterial genes would have been targeted for amplification and sequencing. They would have sent that data back to us, and then using the data suite pipeline, each sequence was then matched in a library to determine the identity of the bacterial species present in each sample. The data was then organized into a single R object using the PhyloSeq package. The goal of this study is to use that genetic information to investigate the co-occurrence patterns in the microbiomes of these 6 species of corals. We hope that by investigating these patterns, we'll be able to find key bacterial species and determine their role in the microbiome and the holobiomes. These key bacterial species fit the definition of what is known as the core microbiome, which in a broad sense is the stable and consistent components across complex microbiome assemblages from similar habitats. The reason why we want to identify the core microbiome is because it is the first step in defining what is a healthy community. In terms of the coral microbiome, this is a term that has eluded coral reef ecologists for a while. We also hope that it will provide direction to predict community responses to perturbation and therefore guide the manipulation of communities to achieve desired outcomes. To do so, we will first use Spearman correlation to determine the relationship between different species for each of the samples. That information will then be used to build a network such as this one, showing the relationships between microbes in soil. Here, each node represents a different genus of bacteria, and the size of the node is reflective of the abundance of the genus, the amount of species in the genus. Each edge or line corresponds to a positive or negative relationship between species. Based on those networks, we can ask further questions such as, how did the network change from 2021 to 2022? And was this change a result of a change in the water source from well water to seawater? And is the microbiome healthier now? The answer to this question hopefully will inform conservationists of the best practices of raising corals in our land-based industries. We could also ask, how similar or different are the networks of different species? And are there any common hub microbes amongst coral species? This would give us an idea of whether there's a core microbiome across species. To achieve these goals, several disciplines will be utilized. Previously, genetics was used to identify and quantify the bacteria. For this study, data science will then be used to build and compare the networks to identify those key microbes. And then microbiology can be used to determine what is the function of those key microbes. Lastly, coral reef ecology must be applied to determine the impact that the microbe has on the coral and whether it will confer higher or lower resilience. Overall, it is hoped that this research can be used to identify beneficial microorganisms that can be used as probiotics to confer greater resiliency to corals. Thank you for your time. I'd like to acknowledge Moat Marine Lab for allowing me to use their data, as well as Dr. Trailer and Dr. Murty for advising me on this project.
Thank you, Shara. Any questions for Shara from the audience?
So I know that we're a little bit over our time. So a great presentation again, like all the others, Sarah. So I have a little bit of a question for you. So, for example, are you expecting that the microbiome composition may change over time and may be contributing to bleaching, or vice versa? Or basically, you know, if you change the microbiome, may become more resistant to bleaching? Is that some parts of the overall, you know, longer term goals, I guess?
Yeah, so the longer term goal, so we only have 2 years of data, but the longer term goal with this data is to build a model to determine what is the healthy microbiome. And then we could put in like perturbations to see what would happen if we change the amount of this bacteria or if we add in a new bacteria. Because that's what happened out in the ocean. But we can't sample all of those because one is too expensive and two is just it's too much out of the way. So yeah, we're trying to see it's both. Yes, we're trying to see what happens when we change things, but also what is the ideal and how could we make those ideal microbiomes and probably inoculate corals with it so they are more resilient.
It's on, I think, no?
Hello? Okay.
Hello, yeah.
That's a great presentation. Thank you. When you're doing the genetic analysis of the microbiome and doing, I'm assuming PCR, to identify the different genes, are you controlling or do you have a mechanism to control for the potential prevalence of very abundant bacteria versus the one that might be a little bit more rare in terms of population, because it's going to be a mixed sample?
Did you hear the question, Shara? Oops.
Yeah, the question was,
I'm so sorry. Could you repeat that? I can't hear.
Sure, no problem. So when you're taking the samples for the microbiome and running a genetic analysis, are you controlling or at least taking into account the potential abundance or overabundance of some microorganisms versus ones that might be more rare to help identify what might be or detrimental versus beneficial to the organisms, to the coral?
Right. So that has been a problem in coral microbiology for a while. And one of the ways they accounted for that when you're looking at abundance data alone is to use relative abundances, but that wouldn't work in the case of network analysis. So I was looking into some other ways to control for it, and there are like a variety of things you could use, but all of them have their advantages and drawbacks. So that's something to look into as things go forward, but that's a really good question.
Thank you.
All right. Am I muted? Somebody has a question from? Okay. All right. Well, in the interest of times, I think we should probably come to a close. Thank you all for coming, for applying, for putting all this thing together. So the fellows will meet twice more before actually presenting their final results in April of this year. I encourage you to reach out to your mentors, get your mentors and advisors in the same room to hash out ideas, and try to get bi-weekly meetings so that you make constant progress. Time goes very quick, right? Especially when you're in the initial stages. All right. Thank you very much. Thanks, Shara.
Thank you. Bye.
Bye-bye.